We are working in the lentiviral vector space for sickle cell disease and eventually beta thalassemia. Aruvant's mission is to use these transformative therapies to treat severe blood disorders. We have an ongoing phase 1/2 program in sickle cell disease, and that is called ARU-1801. It's an ex vivo lentiviral vector process, and we're actively enrolling patients with sickle cell disease. This same vector, same program can be applied to beta thalassemia as well. As far as our strategy and what differentiates us relative to others in the sickle cell space is we have a differentiated payload. And I'll tell you more about why we believe fetal hemoglobin is the best payload. But we believe that its anti-sickling properties and its increased oxygen affinity are the proper payload to cure this disease. That more potent payload allows us to get by with reduced intensity conditioning. Reduced intensity conditioning opens up just enough of the bone marrow space to get gene engraftment and expression, stem cell engraftment and gene expression, but it doesn't come with near-term adverse events from longer-term neutropenia and cytopenia. This would enable us to have patients out of the hospital more quickly and potentially even dosing them on an outpatient basis. This is not a new program. Aruvant as a company is new. It is a subsidiary company of Aruvant Sciences and is based on technology from 2 decades of research by Dr. Poonam Malik at the Cincinatti Children's Hospital. She's been at this design for quite a while and optimizing the payload, promoters, the sequencing of the actual wild-type gamma globin, which codes for fetal hemoglobin. And a lot of changes over time have already optimized and got us into the clinic. Under her management, she dosed 2 patients back in 2017. So we have 2 years follow-up on the first 2 patients, and we have more patients consented, enrolled, and we hope to dose them later this fall into winter. As far as 1801, what actually is our program? It's an ex vivo lentiviral vector. We're using plasmids to form those lentiviral vectors. That work is currently being done in Cincinatti Children's Hospital. Those lentiviral vectors are then inserted into the patient's own autologous stem cells. We can do that both through bone marrow aspirate as well as mobilized peripheral blood in order to gain access to those cells, and those cells, post-modification, are reinfused back into the patient. The 2 differentiating factors that we're excited about our program is one is the superior gamma globin gene, and the second is the reduced intensity conditioning. As far as the payload itself, why is fetal hemoglobin the right approach for this disease is because fetal hemoglobin by nature is a better hemoglobin in order to ward off the sickling of red blood cells. In the formation of the tetramer that is hemoglobin, even a mixed tetramer that has one component of fetal hemoglobin does not sickle, versus an adult mixed tetramer still has a propensity to sickle and thus still have symptoms and side effects. Additionally, oxygen affinity is important. The red blood cell sickles when it drops all of its oxygen out at the capillaries, out at the cell site. And by going from a sickle oxygen affinity curve back to a more normal curve is where you then don't drop all of the oxygen and don't get into a polymerized state where the red blood cell is sickling. We believe that with our mixture at 1.5 to 2 times oxygen affinity, we don't have to get to high levels of fetal hemoglobin expression. Potentially as low as 15 to 20% is enough to ward off sickling of red blood cells. To get a little bit more into the payload, this is not just wild-type gamma globin, but there was a particular edit made by Dr. Malik in her lab years ago that focused on the glycine position at position 16, switching to aspartic acid. And that's where we come up with the G16D notation for our HbF. In the bottom left, you'll see a graphic that shows that because we're not silencing the background rate of sickle, we need to outcompete, our gamma globin needs to outcompete those sickle globin in order to form healthy HbF. The G16D position is at the interface of the alpha globin and the beta or gamma globin, and it's an increased affinity to make that matching between the two proteins. In two different mouse models, in the bottom right corner, you'll see both in a Berkeley mouse model, which is a mixed model of sickle cell and beta thal, as well as in a Townes mice knockout model, which is more pure sickle cell model, we see 1.5 to 2-fold increases in the amount of HbF produced controlled for vector copy number. So this is meant to be controlled for the amount of protein produced. We're seeing 1.5 to 2-fold increases in the amount of formations of the tetramers that are hemoglobin. This is important so that even at low levels of gene expression, we're seeing a large amount of actual formation of beneficial anti-sickling hemoglobin. That enables us to go with reduced intensity conditioning. A lot of lentiviral vectors and a lot of gene editing, gene therapies that you have heard from use busulfan. Busulfan is a highly ablative chemotoxic agent that has its own corresponding side effects. We are going with a reduced intensity conditioning that frees up just enough space in the bone marrow for our autologously modified stem cells to engraft. This can be done at lower levels of chemotherapy. This reduces near-term side effects. We see both cytopenia and neutropenia, so both platelets and white blood cells recover much more quickly post-dose than we do see with busulfan in other trials. Additionally, our therapy has potentially lower risk of infertility. We've only dosed 2 patients, so we don't yet have proof in our trials, but we have seen this agent used in AML and in other settings with women that were able to bear healthy children following doses of the conditioning agent. And then also there's potentially lower risk of secondary malignancies, and this is something that we hope to monitor for all of our patients over a 15-year follow-up period. In addition to the patient improvements from reduced intensity conditioning, there's also improvements for the point of care. Fewer days in the hospital, potentially even eventually getting to an outpatient setting. Greater translatability. If you don't need a high intensity, almost ICU-like environment, you could take this therapy to other points and take this to many other clinical settings, both in the US, Europe, outside of the major academic centers, as well as potentially centers in Africa and India where there's a predominance of sickle cell disease. We're very excited by the dramatic clinical improvements that we've seen in 2 patients to date. This is a data cut that we shared at ASGCT last May in Washington, D.C. For patient 2, at that point in time, we had 21 months of follow-up, and for patient 004, we had 15 months of follow-up. Both of those patients continue to do well. Let me just walk through this data a little bit to show you why we're so excited about this clinical improvement. As far as hemoglobin levels, anti-sickling properties are the important component, and we believe that 29% in the first patient and 20% in the second patient are sufficient. We're seeing meaningful clinical improvements in vaso-occlusive crises. Both of these patients were on the severe end of the spectrum, with 48 vaso-occlusive crises in the historic period of 18 months prior to therapy for patient 002, and for 20 events in the 18 months for 004, and that's gone way down to 2 and to 0. Additionally, this better management of the disease has allowed these patients not to have to be hospitalized for any acute events, disease-related events, and so that's gone from 13 to 0 in the first patient, and it's a harder number to measure in the second. They were in a daycare clinic setting, but that has also gone to no hospitalizations. Additionally, we're looking at quality of life improvements. One of the quality of life improvements that we've measured so far in these 2 patients was their ability to discontinue opioid usage. And both these patients are very excited that they no longer have the haze of daily opioid use that they were kind of under when they were taking opioids to get by of the pain of their vaso-occlusive crises. As far as where we're at in our ongoing development, we have more patients consented and enrolled into our protocol. We've enhanced the protocol, taking it over from Dr. Malik and the Cincinnati Children's Hospital to our sponsored protocol, and we hope to treat one patient per month, really starting later this fall throughout the winter into the spring, eventually expanding beyond that current phase 1/2 to enroll other clinical sites. We have talked with the FDA, and we hope to talk again with them about quickly moving into a pivotal setting. As with most gene therapies, we think a small sample size of patients, potentially on the order of 20, 25, 30, is enough to prove that this therapy is working well and benefiting in an area where there's a lot of unmet need. And we're working with the FDA to gain alignment on that pivotal clinical study protocol. We are also transitioning out of the Cincinnati Children's Hospital. They are producing our viral vector batches as well as doing our cell processing, and we're working with 2 well-known CDMO partners to do viral vector manufacturing and cell processing at a much bigger scale, and getting that process set up such that all of our pivotal patients are treated with our commercial scale process. We are currently active in sickle cell disease. We hope to switch that very quickly to a pivotal program in sickle cell disease, and potentially within 12 to 18 months get that moving. And then this exact same vector and protocol can be used for transfusion-dependent beta thalassemia. And so that's a program that we look to expand into also in the near future. Thank you for your time today.